In this video, we're going to complete our discussion of glycolysis. So, if you recall, glycolysis starts out with glucose. Then through a series of steps, it is ultimately broken apart into two 3-carbon molecules, which are called glyceraldehyde 3-phosphate. The first 5 reactions of glycolysis are the energy investment phase because they require the hydrolysis of 2 ATP molecules in order to occur. Now here we're going to pick up with our 2 glyceraldehyde 3-phosphate molecules, and we're going to follow their conversion into 2 molecules of pyruvate. These last 5 steps of glycolysis are considered the energy generation phase because we will produce 4 molecules of ATP. So it's important to remember one thing when we're going through here. We're going to be looking at the conversion of, say, glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate. That step we're going to show at once, but it's occurring twice for every molecule of glucose because, again, we have 2 molecules of glyceraldehyde 3-phosphate. So let's get started with step 6 of glycolysis, where we're going to take that glyceraldehyde 3-phosphate that was produced, and again, we generate 2 of these for every molecule of glucose, and we're going to convert the glyceraldehyde 3-phosphate into a molecule called 1,3-bisphosphoglycerate. So essentially, we're going to be adding a phosphate group onto this in order to convert it to our 1,3-bisphosphoglycerate. The enzyme that catalyzes this reaction is glyceraldehyde 3-phosphate dehydrogenase. This reaction under standard conditions has a slightly positive delta G, but again, under cellular conditions, that delta G is going to be closer to 0, so it's going to be able to be reversed. It's going to be slightly negative depending on the cellular conditions. So let's take a look at this reaction in a bit more detail and also the enzyme that catalyzes it. So glyceraldehyde 3-phosphate dehydrogenase is our enzyme. And if we look into its active site, here we're blowing the active site up a little bit, it has a histidine, which is going to be a catalytic residue, and we have a cysteine, which also is going to participate in catalysis. We have a covalently bonded molecule called NAD+. So we want to talk about that, introduce that to you here in just a second. Okay, but a few more notes about this reaction and this enzyme. This is the only reaction in glucose that uses inorganic phosphate. Again, inorganic phosphate is just a phosphate by itself, a free phosphate, and that phosphate does not come off of ATP. The reaction generates a high energy anhydride 1,3-bisphosphoglycerate. So 1,3-bisphosphoglycerate is the compound that's generated, and it is a very high energy compound. I'll say more about that in a second. And as I mentioned previously, this enzyme has a tightly bound NAD+, which is regenerated by a mobile NAD+. So make a note of that, and we'll discuss it more here in just a second. Okay? So let me get to this high energy compound. If you recall, we've looked at this table in the past. We have the hydrolysis of ATP releases 32 kilojoules per mole. And here, look at this 1,3-bisphosphoglycerate, the product of this reaction. When it is hydrolyzed, which occurs in the next step, as we'll see, it releases 49.4 kilojoules. So this 1,3-bisphosphoglycerate is a very, very high energy compound, and therefore it can be used, that energy of hydrolysis can be coupled to make ATP, and we'll look at that in the next step. Okay? Now, we said that our enzyme has a tightly bound NAD+ molecule. Let's look at that just a little bit. NAD+ stands for nicotinamide adenine dinucleotide. If you look at the structure down here, here's the NAD+, and it has, if you remember the structure of ATP, we have a nucleotide or a nucleotide here and a nucleotide here. So it's dinucleotide. It has two nucleotides, and then it also has, I've kind of covered it up here, but if you look here, this is nicotinamide, which is derived from the vitamin niacin. Now you don't need to memorize the structure of NAD or NAD+, but just have a general idea of its overall structure. Now, what is the role of NAD+? It has a very important role, and we're going to see this a lot in metabolism. It is an electron acceptor. If you look here, the hydrogen, we have one hydrogen here. Over here in its reduced form, it has two hydrogens. So this NAD plus accepts a hydride ion. It accepts a hydrogen along with two electrons. So hydride, the hydride ion has two electrons. And so it is basically NAD or NADH is a hydrogen or electron carrier. It takes electrons off of something, and therefore it is reduced because it's accepting or gaining electrons. And those electrons can be stored and taken on to another cycle or another pathway, which we'll see is oxidative phosphorylation. So NAD plus is the oxidized form. When it's reduced, we generate NADH, and that reduction occurs because we accept a hydride, which is a hydrogen with two electrons. Okay. So let's take a look at the overall mechanism of glyceraldehyde 3-phosphate dehydrogenase. Now, I'm not going to ask you to memorize all the steps, but again, there are a few important things that we do want to be very familiar with. First of all, as we said, here we have a cysteine, and let me back up here just a second. If you recall, we have a cysteine in the active site. We also have that histidine in the active site, and then the NAD+. So let me come back here. So here is that cysteine. Here is that histidine, and this is my glyceraldehyde 3-phosphate. This would be my substrate. Now we start out, we have our histidine is going to pull off an electron from, a hydrogen from my cysteine, and then we're going to get a nucleophilic attack on that carbon here, which is fairly electronegative. So we end up with a covalent bond, covalent catalysis between the cysteine and what was the glyceraldehyde 3-phosphate. Now at this point, the NAD+, which is also in the active site, is going to pull off the hydrogen and those two electrons, which are seen in that chemical bond there. So that hydride is coming over here, and that's going to reduce the NAD+ to NADH. And these electrons come over here, and at this point, we form a thioester. In other words, we've got a carbon-oxygen double bond, but instead of an oxygen here, which would make an ester, it's a sulfur, and therefore that makes a thioester. Now this step right here, where we reduce the NAD+, you can see, remember, we pull off that hydride and we undergo reduce the NAD+, that is very exergonic. Okay, that's a very exergonic reaction. And this part right here where the phosphate joins on, that's fairly endergonic. So essentially we're coupling these two reactions, an exergonic and an endergonic reaction, in order to get that. Because normally, again, the addition of a phosphate group is very endergonic. It takes a lot of free energy to add that phosphate. That's why generally we add phosphates by coupling them through the hydrolysis of ATP or something like that. So we're coupling this exergonic step with the endergonic step right side by side. Now that phosphate joins on here, and then essentially, again, we won't go through all the steps here, but we cleave that bond and we end up with our 1,3-bisphosphoglycerate, where we added the phosphate on here, and that came from an inorganic phosphate. So that's important to appreciate because it's a very unique step. There our cysteine and our histidine are regenerated, and here you can see the mechanism involves a 2-electron transfer. There we're talking about that hydride ion to the NAD+, as well as the formation of an acyl phosphate, which is 1,3-bisphosphoglycerate. So remember, we mentioned that we're forming this molecule here, which upon hydrolysis has a very large negative delta G. Now, just to kind of reiterate what we were talking about before, we have an aldehyde, and if we transfer a hydride over here to the NAD+, so the NAD+ is reduced, we form this in water. In the presence of water, we form an acid. Essentially, that's kind of a very summarized step of what we we're looking at before, but that's a very exergonic step. Okay? Now that's coupled to the addition of that phosphate here, and that's a very endergonic step. So we're coupling a very exergonic step with an endergonic step right within that same enzyme. So that's why this inorganic phosphate's able to be added instead of coupling, as I mentioned before, to the hydrolysis of something like ATP. So let's move on to our next reaction, reaction 7. We take that 1,3-bisphosphoglycerate that we said many, many times here is a very high energy compound, and we break it down into 3-phosphoglycerate. So we pull off a phosphate and we add it to ADP. Now this is a very important step because we are generating ATP at this step. Okay? So we're pulling off a phosphate and we're actually coupling this removal of the phosphate to the phosphorylation of ATP. So removing this phosphate here, we said the delta G for that was about negative 49 kilojoules per mole. The formation of ATP from ADP is going to be about positive 32 kilojoules per mole. So overall, we still have a very negative delta G, about negative 17.2. This enzyme that catalyzes this reaction is called phosphoglycerate kinase. It's a kinase because it is transferring a phosphate onto another molecule, in this case from our 1,3-bisphosphoglycerate to ADP. So this is what's called substrate-level phosphorylation, where we're directly phosphorylating the ADP to ATP. Now we will see that there is another type of phosphorylation called oxidative phosphorylation, and we'll look at that when we get into the electron transport chain, but this is a term that describes when you directly phosphorylate something like ADP from transferring it directly from another molecule onto ADP. So here you can see we have our glyceraldehyde 3-phosphate to form 1,3-bisphosphoglycerate, and this overall reaction is somewhat exergonic. This was the step that we looked at previously, reaction 6. And this is essentially coupled to this reaction, which has a very exergonic. So here, we're not just coupling, reactions with steps within the same reaction. These two steps, step 6 and step 7, can be thought of as being coupled together. So this step here, step 7, is essentially driving step 6 along and getting it to occur, even though it's not thermodynamically favorable, at least under standard conditions. So let's look at reaction 8. Here we have our 3-phosphoglycerate, which we just produced, and we are going to convert that into 2-phosphoglycerate. Now what we're doing here is essentially just moving a functional group from one carbon to the next, and an enzyme that does that just transfers we have an internal transfer of a phosphate group is an enzyme called a mutase. So this would be phosphoglycerate mutase. Now essentially what we're going to do here is we're getting this molecule ready to form a very high energy molecule where we can make some more ATP, and we'll see that in step 9. But we're basically priming it, getting it ready to form a high energy compound. So this mutase is transferring the phosphate from one carbon to the next. Now you may look at this and say that think that the enzyme simply pulls this phosphate off and transfers it over here, but it's actually a little bit different than that. Let me show you that what we're talking about, how this enzyme does this reaction. So as I mentioned, a mutase is a class of enzymes where it has an internal shift of a functional group or internal transfer of a functional group. And this reaction proceeds through an intermediate 2,3-bisphosphoglycerate, and it is reversible. Now, again, let me explain what we mean by this in just a second here. The mechanism involves the phosphate transfer from a histidine onto the enzyme at the C2 Then this histidine removes the phosphate from the C3 of the 2,3-bisphosphoglycerate to form 2-phosphoglycerate. Okay, now that's a mouthful there, I know. So let me show you this in a bit more detail to try to make some sense of what we're talking about here. Okay, so our enzyme already has a phosphate attached to a histidine. So our enzyme has a histidine in the active site which has a phosphate attached to it. And then our substrate, the 3-phosphoglycerate, comes into the active site. That enzyme transfers the phosphate that it already has attached there onto our 3-phosphoglycerate at the 2-carbon. So now we have the enzyme with just the histidine, no phosphate, and we have 2,3-bisphosphoglycerate. Then what the enzyme does is that histidine, which no longer has that phosphate, it pulls that phosphate off of the 3-carbon and transfers that back to the histidine. So we essentially have our enzyme regenerated with the histidine and the phosphate attached to it, and we have 2-phosphoglycerate. Now, an important note about 2,3-bisphosphoglycerate, you may think that hopefully that sounds a bit familiar. That is a very important allosteric effector of hemoglobin. So we've talked about that previously. So let me show you just one more figure because this can be a little confusing, showing this phosphoglycerate mutase in action. So we start out here. This is my 3-phosphoglycerate substrate, and we have our histidine. Here's my enzyme's active site, and we have a histidine there, which already has a phosphate attached to it. That phosphate is transferred to that 2-carbon. So here now we have that the histidine has transferred its phosphate to that 2-carbon, the 2nd carbon, and now we have 2,3-bisphosphoglycerate intermediate. Then the enzyme, that histidine, pulls the phosphate off, but not, of course, the same one. It pulls it off the 3-carbon, and that generates our 2-phosphoglycerate and regenerates our enzyme. So let's move along here and look at our next steps, reaction 9. Here we have our 2-phosphoglycerate, and that is converted into a very high energy molecule called phosphoenol pyruvate. The enzyme that catalyzes this reaction is called Enolase, and this is phospho because we have a phosphate group, enol because we have a carbon-carbon double bond, so an alkene, and then the pyruvate is the overall name of this. So phosphoenol pyruvate is the molecule that's produced by a catalytic reaction with enolase. Okay. Then we move on to our final step in glycolysis, reaction 10. This reaction is going to be catalyzed by the enzyme pyruvate kinase. And in this reaction, we will generate another molecule of ATP. Now remember, everything here is occurring twice because we started out with 2 molecules that are passing through here, 2 molecules of glyceraldehyde 3-phosphate. So here we have our phosphoenol pyruvate, which again is a very high energy molecule. If you look back at that table of high energy molecules that we showed you previously, phosphoenol pyruvate is at the top of the list. It's one of the highest energy compounds that we encounter a lot in biochemistry. This molecule takes this phosphate and adds it onto ADP, so we have a substrate-level phosphorylation occurring to produce phosphate and ATP. Now this reaction, even though we are phosphorylating ADP to form ATP, which requires a lot of free energy, because there's so much free energy released, this step is very exergonic. So essentially, this last step kind of drives the whole pathway in an exergonic spontaneous direction. A couple of things about pyruvate kinase. Although the synthesis of ATP is endergonic, again, the overall reaction is exergonic because the breaking off of that phosphate, the hydrolysis of phosphoenol pyruvate is very, very exergonic. So that is the steps of glycolysis and some important points about the different enzymes. We want to be sure we know the substrates, the products, the enzymes which catalyze those reactions, and also some of those interesting facts that we looked at. So let's just go through it real quick one time just to sum up. Remember, we start off with glucose that enters the cell. Generally, this occurs a lot in the liver. Glucose is converted to glucose 6-phosphate by hexokinase. This requires ATP. Then we convert glucose 6-phosphate to fructose 6-phosphate. This is glucose 6-phosphate isomerase. Fructose 6-phosphate is converted to fructose 1,6-bisphosphate by phosphofructokinase. This requires a second ATP. Remember, this step right here is one of the most important regulatory points in glycolysis, because this is the first committed step in glycolysis. Then our fructose 1,6-bisphosphate is broken apart. We form one glyceraldehyde 3-phosphate, one dihydroxyacetone phosphate. The dihydroxyacetone phosphate that's formed is converted into glyceraldehyde 3-phosphate, and that enzyme is triose phosphate isomerase. So we end up with two of these glyceraldehyde 3-phosphates. Those two glyceraldehyde 3-phosphates are then converted into 1,3-bisphosphoglycerate. Again, we've got two of those. This step produces NADH. Again, two of those, one per molecule. The 1,3-bisphosphoglycerate is converted into 3-phosphoglycerate, and we get our first substrate-level phosphorylation to produce ATP. That's converted into 2-phosphoglycerate by phosphoglycerate mutase. And then Enolase comes in and converts that to phosphoenolpyruvate. Again, a very, very high energy compound. That hydrolysis of that molecule is used to produce our second two molecules of ATP. Again, one molecule per phosphoenolpyruvate, which generates our final product in glycolysis, which is pyruvate. The pyruvate then undergoes a number of different fates. It can go on through into the citric acid cycle and oxidative phosphorylation, or it can undergo enter a couple of other pathways, which we'll discuss at a later time.